Hello, everyone. Welcome to another session of BioCool classes. Today's topic is photosynthesis. And so this is the BioCool class Telegram group. As I said, today's topic is from the chapter Life Processes, Photosynthesis. So in this class, we will try to cover all the important topics which are required for the photosynthesis chapter. This will help in students from CBSE, ICSE, even from the IB board. So these topics are common. So let's proceed with this chapter. Okay. So photosynthesis, one thing we have to understand that this is called autotropic nutrition. Now, the word autotropic means self, auto means self-production, and tropic means food. So autotropic nutrition happens in case of plants. And so the plants, they are called the autotrophs. Why they're called autotrophs? Because auto means self and trop means food. So basically, plants can make their own food. So that's why they call autotrophs as compared to the word heterotrophs. Heterotrophs means those which require ready-made food. So heterotrophs examples are the cow, the man, okay, I mean the human beings. Then we have tiger, lion. They're all heterotrophs because they need food from the plants. They rely on plants either directly, just like a cow, goat, any kind of herbivores, or indirectly, just like a lion, a carnivore is indirectly dependent upon the autotrophs for their nutrition. So now, so autotropic nutrition, what we need to know about autotropic nutrition? It's just okay. I hope the screen is clear. So one thing we need to know that carbon and energy, these two things are required by autotrophs. So autotrophs, they need carbon and energy. And this carbon and energy, these two things are fulfilled by the process of photosynthesis, because carbon and energy is something all living beings, they need this. Plants, being a living being, they also need carbon and energy. And this carbon and energy of the plants is fulfilled by the process of photosynthesis. So plants, they take, so autotrophs or plants, they take substances from outside. So autotrophs, they're taking substances from outside to fulfill this carbon and energy. How? So what are they taking? They're taking the raw materials like carbon dioxide. They're taking raw materials like water. So carbon dioxide is taken from the atmosphere, from the air. Water is taken from the soil. And then they convert this into carbohydrate. They convert this into carbohydrate, which we write as C6H12O6, that is the glucose. And by utilizing sunlight, So sunlight or the radiant energy that provides the energy for carrying out this whole process. And then we have chlorophyll. Chlorophyll are the pigment molecules which help in capturing this sunlight and converting the sunlight into chemical energy. So from radiant energy, it is converted to chemical energy. And so, as I said, ultimately carbohydrate is formed. This carbohydrate is utilized for providing energy. So plants, they make this carbohydrate this in the form of glucose. So the glucose is broken down, and from this glucose, energy in the form of ATP is derived. The plants, they need some of the glucose for their own physiological processes. Rest of the glucose is stored in the form of starch. So plants, the excess glucose that is made in photosynthesis is stored in the form of starch. And so in the plant's leaves, we see starch present. Okay. In case of animals, so animals, they get carbohydrate from the diet. So this is happening in case of animals. Just a comparison. So animals, they're getting the carbohydrate from their diet because animals are heterotrophs. And this diet, then they carry out digestion. After digestion, this carbohydrate from a complex carbohydrate, it is converted into glucose. This glucose is then absorbed at the small intestine, and then this glucose is utilized, and from here ATP is derived. This process is called assimilation. This process is called assimilation. The excess of the excess glucose, however, is stored as glycogen. Is stored as glycogen where? In the liver of our body. So just to give a comparison, plants, the excess glucose is stored as starch in the leaves. In case of animals, the excess glucose is stored in the form of glycogen in the liver. Whenever the animals, they need it, they break down this glycogen and they convert it into the glucose if they need any extra energy. Okay. So that's what they say here in this part of the notes, that in animals, it is stored in the form of glycogen in humans and in other animals. Now, so next, this is the equation of photosynthesis. So where we see 6 molecules of carbon dioxide. So this is the carbon dioxide we're getting from the air or the atmosphere. And we have 12 molecules of water. This is again we're getting from the soil. And we see that how chlorophyll, the pigment molecule, is required to capture the sunlight. And here, as I said, sunlight is the radiant energy. And it's forming C6H12O6, which is basically the glucose, 6 molecules of oxygen. This is the oxygen that is coming out of the plants during photosynthesis. And this is the oxygen which all aerobes, it means those which carry out aerobic respiration. Aerobic respiration. Okay, so the aerobes, they need this oxygen. We human beings are an example of an organism which carries out aerobic respiration. So we need this oxygen. We carry out cellular respiration. We carry out cellular respiration with the help of this oxygen. And also there is formation of water molecules. So this is the complete equation of photosynthesis. 6 molecules of carbon dioxide, 12 molecules of water in the presence of chlorophyll. Sunlight provides the energy. There is production of glucose. There is production of oxygen. And there is also production of water. Now, we need to look at where this whole photosynthesis is happening, where this is very, very important. It is happening obviously in the green leaves. So now we will look at the cross section of a green leaf. Look, this is a green leaf, and as you can see, this is the midrib of the leaf, and you can see the venation in the leaf. This is called the leaf blade or the lamina. So we can look at another detailed picture. And as you can see in this picture, so in this picture, we can see one by one. This shows the whole process of photosynthesis in leaves. Now, we just zoom this part. And so this is a leaf, and we have taken we're just zooming this leaf. As we zoom this leaf, you can see this is the midrib of the leaf from where the water is coming through the xylem vessels. And look how sunlight, the leaf is receiving sunlight. Now, we have taken a small cross section here. Okay? We'll take a small cross section here, and this we are magnifying the cross section. So let's magnify this more. So now we can see this is basically a three-dimensional picture of the leaf. What do we have here? We have the upper epidermis. This is the upper epidermis. Okay, the epidermis. These are the cells of the epidermis. And as we know, the upper epidermis, it protects the leaf from different kind of microbes. Okay, and also sometimes there is a cutin layer. Okay, there is a cutin layer or cuticle layer on top of this upper epidermis, which protects the leaf from any kind of evaporation also. Now, beneath the upper epidermis, we have these cells which are called the mesophyll cells. So you see we have these kind of elongated cells, and we have the round cells. So these elongated are called the palisade cells. So they're all mesophyll cells, both the palisade, and this is called the spongy cells. So they are type of parenchyma. They're a type of parenchyma. Parenchyma is it's a permanent tissue in plants, and parenchyma is present everywhere in the plants. So palisade is a type of parenchyma. Spongy is also a type of parenchyma, and parenchyma has this kind of characteristics. There is a lot of intercellular space. That is the special characteristics of parenchyma tissue. And parenchyma tissue which carry out photosynthesis is known as chlorenchyma. So parenchyma tissue that carries out photosynthesis, because they have pigments called chlorophyll. So this is called the chlorenchyma. It is a special type of parenchyma, chlorenchyma. So mesophyll cells are basically a type of chlorenchyma. Mesophyll cells are two types. We have palisade cells and we have spongy cells, depending upon the shape. So now here we can see these are the xylem vessels. You can see this is basically called the bundle sheath. The bundle sheath has the xylem vessels. It also has the phloem, which carries the food, right? So xylem vessel carries the water from the soil, water and minerals by the process of ascent of sap. And then we also have parallelly running with the xylem is the phloem. As you can see, this is the phloem. This is the xylem. They run parallel. The phloem brings or carries the food that is made by photosynthesis in the leaves to different parts of the plant. So phloem is transporting or translocating, that's the word. It is translocating food that is made in the leaves to different parts of the plant, especially the non-green parts of the plant. Okay, and xylem is transferring water and minerals where to these mesophyll cells so that photosynthesis can take place. All right. So now, as we move to, So here, this is basically now so now we go back to the previous figure. So this is one mesophyll cell. This is one mesophyll cell. We can see how the mesophyll cell has this large vacuole in the middle, and the vacuole of a plant cell has cell sap. These green round things are called the chloroplast. So they are the chloroplast here. This is called the chloroplast. This is the vacuole. You see the cytoplasm. This is the cell wall outside. Okay? And so this is the chloroplast. And now what we have to see here is how the carbon dioxide and the water are reaching this mesophyll cell. So I said the xylem vessel is transporting the water, but how is the carbon dioxide reaching the mesophyll cell? So let's look at this picture one more time. Let me okay. So now, if we can look at this picture, we see how the carbon dioxide is entering through the stomata. You see, these are on the lower epidermis. We can see this kind of the stomata. Stomata are basically pores, and these pores, they're allowing gaseous exchange into a leaf. You see, this is the stomata. As we magnify this, and the stomata is guarded by two cells called the guard cells. And we see how this carbon dioxide is coming and entering into the leaf, and how ultimately it is reaching into the chloroplast, right? So now this is another picture of the leaf cross section, and you can see the upper epidermis. You can see the bundle sheath here, and now we know the bundle sheath is made up of the vascular bundles, that is the xylem and the phloem. This is how the chloroplast looks like. It is a discoidal shaped uh, organelle of the plant cell. So what I mean by organelle is that organelles, so plant cells are eukaryotic cells. They have different kind of organelles like the nucleus, like the mitochondria, like the chloroplast. There are peroxisomes, there are the Golgi, there are the endoplasmic reticulum. So chloroplast is also a type of organelle which stores the pigment chlorophyll to carry out photosynthesis. So now, when we look at the chloroplast, so now we know the exact location of the chloroplast inside the mesophyll cells. This is another picture of a 3D picture of, this is another three-dimension picture of the leaf, inside of a leaf, and we can see the upper epidermis, and you see there is a cuticle on the top of this, and we have the two types of mesophyll cells, the palisade and the spongy parenchyma, and we can see the guard cells, which is guarding the stomata, and how from here the carbon dioxide is entering and it is directly reaching into the mesophyll cells and into the chloroplast. So now this is another picture, a microscopic picture. This is a light microscopic picture and of a leaf, and we can see how each of these rectangles are mesophyll cells, and we can see how they're packed with chloroplast. So mesophyll cells are the photosynthetic cells of the plant. Now, this is a picture of a chloroplast. Let's just magnify this, and we can see the chloroplast has an outer membrane and it has inner membrane. So let's just point out. So this is the outer membrane. It is so chloroplast is an organelle which has two kinds of membranes, the outer membrane and the inner membrane. So this is the inner membrane. This is the outer membrane. Between the two membranes, we have this intermembrane space. What else we have? In the chloroplast, there are these kind of disc-like structures which are called the thylakoids. You see, the thylakoids. The inside of the thylakoid is called the thylakoid lumen. So this is the thylakoid lumen. So this is the lumen or the inside of the thylakoid. These thylakoids are arranged as stacks of coin, just like coin stacks. You take many coins. So this stack is called the granum. So this is called the granum. So one stack is called granum, many stack is called grana. So this is the singular. This is the singular, and plural is the grana. So many stacks are called the grana. You can see between two granum, there is this connection, which is called the lamella. It is also called the stroma lamella, either lamella or we can call it the stroma lamella. Why stroma lamella? Because this aqueous fluid that is surrounding these grana, it's called the stroma. So stroma is basically an aqueous fluid, and it is on the stroma we have these grana are present. The grana and the stroma lamella, they're basically floating on the background matrix that is called the stroma. It is in the stroma we have the different kinds of enzymes, that are required for photosynthesis. One of the enzyme example is Rubisco, and we will learn this Rubisco enzyme is required in a process called Kelvin cycle, which is also called the dark reactions of photosynthesis. So more, we want to look at the chloroplast from a different angle. So this is the chloroplast, which is again, it's an organelle, it's a plant organelle. And you can see that how the chloroplast has these outer membrane. This is a different kind of perspective. So outer membrane, and then we have the inner membrane, and in between we have the intermembrane space. All right. We have these stacks of thylakoids, stacks of thylakoids known as the granum, and the entire, so more than one granum is called the grana. Granum, plural is grana, and connected by the lamella or stroma lamella. And you see this is this aqueous thing here outside, this matrix or this background is called the stroma. In the stroma, all these membranes are floating in the stroma. Okay. All right. So now we go deep into the thylakoids. So now we are concentrating on these thylakoids, each of these thylakoids, and we look at the thylakoid membrane. So the thylakoid membrane is a lipid bilayer. You see, this is now we are focusing on the thylakoid membrane, and you can see thylakoid membrane is a bilayer, it's a lipid bilayer. Okay, so this is basically a lipid bilayer where this has the polar head and we have the tail. So we know that this is basically the phospholipid bilayer where, so let's just point it out. This is the polar head, and then we have a fatty acid tail, this is the same lipid bilayer which we see in the plasma membrane of a plant cell or an animal cell. So plants, they have a cell membrane which is made up of this lipid bilayer, and also plants have an additional cell wall. Okay, just some extra information. So so coming to that, so this is basically the head, and this is the tail. This is the this is called the phospholipid bilayer. So in this thylakoid membrane, there is embedded this protein. It's a membrane protein. We can call this a transmembrane protein because it is spanning the entire thylakoid membrane. So this is called the photosystem. What do we call this? This is called a photosystem. So there are two kinds of photosystem. We have photosystem 1 or PS1 and photosystem 2 or PS2. And you can see if we study this photosystem, what do we see is the photosystem has this outside area which is called the light harvesting complex. You see the light harvesting complex, and it has this inside area which is called the reaction center complex. So photosystem has two parts, the outside light harvesting complex. Also sometimes it is called the antenna complex. It is also called the antenna complex. And the antenna complex is filled with different kind of pigments. There are these pigment molecules, especially we have chlorophyll B, there is xanthophyll, and we have carotene mainly. So these two are in combined called the carotenoids. They're called the carotenoids. All right. And in the reaction center, we have it is mainly rich with chlorophyll A. Okay. So the plant, we can see in the thylakoid membrane, we have these photosystems which contain the pigments, the photosynthetic pigments. So next time we come across the concept that plant leaves, they have chlorophyll, then we should exactly think that inside the plant leaves, we have these mesophyll cells. Inside the mesophyll cells, we have the chloroplast. Inside the chloroplast, we have these thylakoids. Thylakoids have a membrane, and in the membrane, there is this protein called transmembrane protein, which is called the photosystem. Inside the photosystem, we have again two divisions. We have the outside light harvesting complex and inside reaction center. This is exactly where we have the photosynthetic pigments are present. What are the photosynthetic pigments? The main photosynthetic pigments are chlorophyll A, chlorophyll B. We have the carotenoids, which comprises of xanthophyll, carotene. Okay. We also have other versions of chlorophyll, chlorophyll C, chlorophyll D, chlorophyll E. But in higher plants, we mainly see chlorophyll A and chlorophyll B. All right. So now we move on to the process of photosynthesis. Now, before we move on to the process of photosynthesis, we need to know that there are two kinds of photosystem, photosystem 1 and photosystem 2. And I can show the picture of the two photosystems, how they're arranged in the thylakoid membrane. So this is basically a picture of, look here, we have the chloroplast at the top, and now we are magnifying the membrane of, so we are basically, here we have just taken a small section from the thylakoid membrane and we are magnifying that, we are zooming that. And when you're zooming that, this is exactly how it looks like. And you can see how this is the thylakoid membrane. Again, it's a lipid bilayer, phospholipid bilayer. And we have these embedded protein. We have these membrane proteins, integral membrane protein, we call them. Okay, so we call them the integral membrane proteins. And this, so we have, this is the photosystem 2 and this is the photosystem 1. As I said, these photosystems, they are packed with the pigment molecules, xanthophyll, carotene, chlorophyll A, chlorophyll B. There are also a bunch of electron carriers which are embedded in the thylakoid membrane. This is one electron carrier, here is another electron carrier. And these electron carriers have different kind of names. So in this picture, we can see we have the the PQ or the plastoquinone. This is a type of electron carrier. And then we have another electron carrier called the cytochrome BF6. And then we have a mobile electron carrier called the plastocyanine. Okay. And a little bit far away, there is this ATP synthase, which is an enzyme. And we will learn how ATP synthase helps in the production of ATP, adenosine triphosphate, which is the energy currency of a cell. So going back to the two reactions of the plant. So photosynthesis can be divided into the process of photosynthesis. We can say it can be, so there are different kind of processes happening during photosynthesis. So what are the processes? The processes are absorption of light. So first there is absorption of light energy by chlorophyll. So there is absorption of light energy by chlorophyll. So now we know where exactly the chlorophylls are present. The second step is conversion of the light energy into chemical energy. So light energy is converted into chemical energy. So what is the chemical energy? ATP and NADPH. So these are also called the assimilatory power. Assimilatory power. Why they're called the assimilatory power? Because these two are required for assimilation of carbon dioxide. So they help in capturing the carbon dioxide and converting the carbon dioxide, or reducing, to be exactly, that's the exact biochemical word, reducing this carbon dioxide to form glucose. Okay, so these two are called the chemical energy, and they are coming from the light energy. Light energy is converted into chemical energy, and here chlorophylls, they play a role in this conversion of light energy into chemical energy. How is this happening? By a process called the light reactions. So there is a process called light reactions, and these light reactions, they help in capturing the sunlight and converting it into the chemical energy of ATP and NADPH. So again, so there are three steps of photosynthesis. We have the absorption of light by chlorophyll, conversion of the light energy into chemical energy, and then a very, very important step, the splitting of the water molecules, which is called the photolysis of water. Photo means light, lysis means breakdown. So photo, using light, water is broken down, photolysis of water. And this water is broken down, and from here oxygen is evolved, and also protons, H plus ions. Protons are also evolved. And we will know how these protons, they form a proton gradient, and how this gradient helps ultimately in the in the production of ATP. ATP is one of the assimilatory powers. Okay. Okay. So hydrogen and water. Third step is reduction of the carbon dioxide. Reduction of carbon dioxide to carbohydrates, as I said. Reduction of carbon dioxide, and this carbon dioxide is coming from outside, right? And I just showed the picture of how the carbon dioxide is entering through the stomata, and how they're getting inside the mesophyll cells, and finally inside the chloroplast. So this last step of reduction of carbon dioxide to carbohydrate, it is also called the dark reactions. It is also called the Kelvin cycle. Kelvin cycle, which is named after the creator Kelvin, and also his fellow colleague called the Andrew Benson. So it is technically, actually, it is called Kelvin-Benson cycle, or in short, we can say Kelvin cycle. All right. So next we go and we will study the light reactions of photosynthesis. So as I said, the photosynthesis can be divided into two main parts. We have the two types of reactions. We have the light-dependent reactions, reactions dependent on light, which is also called the Hill reaction, based on the discoverer Robin Hill. So the British scientist Robin Hill, he discovered the light reactions, so it is also called Hill reaction. And the second kind of reaction is the light-independent reaction, which is the dark reaction, which is also called Kelvin-Benson cycle. It's a cyclic reaction, so it's called cycle. Okay, and the light-dependent reactions is basically electron transport chain. I've already given an idea why it is called electron transport chain, because in this light reaction, electrons are carried via these kind of electron carriers like the plastocyanine, plastoquinone, then cytochrome BF6, then we have plastocyanine. So there are electron carriers which are bound to the thylakoid membrane, which are carrying the electrons. So the light-dependent reactions, okay, occur within the thylakoid membranes, and the light-independent reactions, which is called the Kelvin cycle, they take place in the stroma of the chloroplast. Okay, so light reactions happening in the thylakoid membrane, dark reactions happening in the stroma of the chloroplast. One thing we need to know, that the dark reactions are called the light-independent reactions. That means light is not directly required. They are indirectly required. So basically, dark reactions are dependent upon the light reactions, because light reactions, they supply ATP and NADPH, the assimilatory power. Without the supply of ATP and NADPH, the dark reactions will not carry out. So in one way, they are dependent on light, but they're not directly dependent on light. Just like the light reactions are directly dependent on light. So that's why they are called light reactions. Light dependent reactions, they're called the light independent reactions. Okay, so let's go to light reaction and see what's going on. So here is the complete overview of what is happening, and you can see this is the chloroplast. This is the chloroplast, and these are the you can see this is the thylakoids, or the grana, we can say, and how they're taking in light. So, and there is photosystem 2, there is photosystem 1, and how they are actually forming the assimilatory power, ATP and NADPH, and these assimilatory power are required for reduction of carbon dioxide into sugar. Okay. This is the glucose, and you see that once sugar is formed, it is required for cellular respiration. Plants, they break down the sugar and they carry out cellular respiration, which provides the plants energy. Then also this sugar, from the sugar, cellulose is made, and this cellulose is basically a component of the cell wall. Cellulose is a polysaccharide. It's a fibrous polysaccharide, which is a component of the cell wall. Also, the excess sugar, the excess glucose is stored as starch in the leaves. All right. So now let's move on to the light reactions. What do we see here? We see thylakoid membrane. We see the two photosystem, photosystem 1, photosystem 2, photosystem 1. Now, a lot of students might be thinking that why the photosystem, this is the photosystem 2, is named 2 before and then 1 like that. Right? Logically, this should have been photosystem 1, this should have been photosystem 2, but it is based on the discovery. It is based on the discovery. Photosystem 1 was discovered earlier. That's why it's called photosystem 1. Photosystem 2 was discovered later. That's why it is called photosystem 2. Although, if you look at the sequence, photosystem 2 is present before the photosystem 1, right? So sequentially, photosystem 2 is present before the photosystem 1, but we name them as photosystem 1 and 2 based on the time of discovery. One was discovered earlier, that's why it is called photosystem 1. The other was discovered later, right? So now, as you see how light is being captured, it is called light-dependent reaction. And the moment light is captured, very important thing happens. The electrons which are present in the chlorophyll, I said this is the antenna complex, and you can see how the moment they capture the light causes electrons present in the chlorophyll molecules, chlorophyll B especially, and also xanthophyll, also carotene. So all these pigments, they have electrons, and the light causes the electrons to go to a higher excited state, and these electrons are coming out and they are transferred. You see how the light causes the electrons to be transferred from one chlorophyll to another chlorophyll molecule. Okay, so this arrow shows how the electrons are transferred, okay, from one pigment molecule to another pigment molecules, and these chlorophyll molecules are now excited. They are now in a higher level from the ground state. They are excited because they have lost an electron. Okay, after they receive light, they have lost an electron and they have transferred the electron to the next chlorophyll molecule, and in this way, in the antenna complex, you see how the electrons are finally transferred to the reaction center. You see how these electrons, they jump from one chlorophyll molecules to another chlorophyll molecules and enter the reaction center. Now, one thing we have to realize, these chlorophyll molecules are in a higher state, in an excited state. They want to return back to the ground state. How they can return back to the ground state? The only way they can return back is when they get an electron. When somebody donates an electron to them, they will go back to the ground state. And this is what exactly they do, is that they attack water, they lyse water. So they're attacking water here. So they're attacking water molecule and they're splitting up water molecule into oxygen and H plus ion. This is called the photolysis of water. Why photo? Because so this is the photolysis of water, and we can see how this water is basically breaking down, and also there is formation of electrons. This is the whole reason the water is being split by these chlorophyll molecules, and this electron goes back to the chlorophyll molecules. Thus, the chlorophyll molecules, they return back to their ground state by getting the electrons from the water. Now, what happens to the electron which was sent to the reaction center? These electrons, so there are these electrons we write as 2e. This electron is then transferred to a primary electron acceptor. So this is called a primary electron acceptor. Okay. From this primary electron acceptor, the electron is transferred to a electron carrier. This is called the plastoquinone. Okay. And this plastoquinone accepts the electron. It also accepts one hydrogen from the stroma side. You can see this is the stroma side of the chloroplast, right? This is the thylakoid membrane. This is the lumen of the thylakoid membrane. That is the inside of the thylakoid membrane. This is the inside of the thylakoid actually. And this is the outside. This is the stroma side. So from the stroma side, one hydrogen or one proton is coming, and PQ, that is the plastoquinone, is then transferring this H plus to the lumen of the thylakoid. So this picture can be seen more clearly. This concept can be understood more clearly in the next diagram. Here you can see how, in this diagram, we can see how the H plus is being transferred with the help of plastoquinone to the lumen side. So this causes accumulation of H plus in the thylakoid lumen, because you see also H plus or protons are made by photolysis of water. So photolysis is already making H plus. At the same time, every time an electron passes to the PQ is transferring one H plus to the lumen side. So the lumen is getting H plus rich, and gradually a gradient is formed. A gradient, because there is more plus charge inside compared to outside. So there is a H plus or a proton gradient. So we call this proton gradient is forming as the electron is passing. Now, so the electron from the plastoquinone, now it goes to the next electron carrier. This is called the cytochrome BF6. Cytochrome BF6 then passes the electron to the next carrier. This is a mobile electron carrier. That means it can move around. It can move around like that. You see the other electron carriers like the plastoquinone and the cytochrome BF6, they are kind of embedded in the thylakoid membrane, but this guy, the plastocyanin, the PC, the plastocyanin can kind of, there is a mobility, it can move around. Now, if you see the second electron, the photosystem 1, the photosystem 1 is doing the same thing. It is accepting light, and so it is accepting light, and just like the photosystem 2, again there is the same thing happening. Again the pigment molecules, they are losing electrons. So we can go back to the previous figure. You see the similar thing is happening. It's accepting light, and then again the electrons, the excited electrons, are transferred from one pigment to another, finally reaching the reaction center. From the reaction center, it is going to the primary electron acceptor. Now, from the primary electron acceptor, it is going to a different set of electron carriers. So first, we have an electron carrier called ferredoxin, FD. Ferredoxin is transferring the electron to FNR, which is the ferredoxin NADP reductase. This is an enzyme. This FNR or ferredoxin NADP reductase is reducing NADP, which is present abundant in the stroma, into NADPH. So this is a reduction because it's gaining hydrogen, right? NADP is converted into NADPH. All right. And one thing we have to understand is PS1, they do not split water. The question is that how does the pigment molecules from an excited state, they go down to a ground state? They do it by taking the electron from plastocyanin. You see the plastocyanin is waiting here with the electron from the PS2. So now this electron is transferred, and this helps the pigment molecules in the PS1 to return to the ground state. So now, all this time, electron has been, H plus ion, proton has been accumulating in the lumen of the thylakoid. So that causes a proton gradient formation, okay? So there is a difference in positive charge. So there is a formation of proton gradient formation, you see? And this proton gradient, so now these protons have to be returned back to the stroma. They have to be returned back to the stroma. It is very, very important. So they have to be returned back to the stroma. How they return back? Via these huge ATP synthase complex. So this is a very, very big enzyme complex which is embedded in the thylakoid membrane. And you see H plus ions, they're traveling. So there is a tunnel inside this, this ATP synthase. This is basically an enzyme. It's a protein, right? So all enzymes are protein. We know this thing, right? Enzymes are basically protein. So you see how the H plus is going through this tunnel outside into the stroma side. And as this H plus is moving from inside to outside, it creates a force which causes this ATP synthase to rotate like that. So it is just same concept as happening in a hydroelectric power plant. In an hydroelectric power plant, how the water is coming and this water is turning the turbine. In a hydroelectric power plant, the water is falling and this water causes the turning of the turbine, and the turbine generates electricity. The same concept we see here. Here, H plus ion is acting as the water. As the H plus is passing through this ATP synthase, which is acting as the turbine, the ATP synthase, it rotates, and this causes ADP, adenosine diphosphate, to react with an inorganic phosphate P and to form ATP, which is the adenosine triphosphate. You see how this is the ATP generation. So we go and see another picture. This picture shows how this ATP synthase actually looks like a turbine. And as the H plus passes, you see, as the H plus proton passes from H plus rich thylakoid lumen to the back to the stroma, right? We see this causes the turbine to rotate. Rotating of this turbine or the ATP synthase causes ADP and PI to, PI means inorganic phosphate. You see, we write inorganic phosphate as P and a small I, forming ATP. So eventually we have NADPH and ATP are produced. These are called the assimilatory power of the light reactions. So now we'll move to the dark reactions, and we'll see that how this assimilatory power is used to ultimately form the glucose. Okay. So dark reactions, first some information about dark reaction was discovered by these two scientists. This is the famous Melvin Kelvin, after whom the Kelvin cycle is popularly named as. So it was discovered by Melvin Kelvin and his colleague Andrew Benson. So it is also called Kelvin-Benson cycle. And they used radioactive isotope to track all the reactions of the dark reaction. Okay. So what are the steps of Kelvin cycle or the dark reaction? It has three main steps, very, very important. Again, dark reaction is happening in the stroma of the chloroplast, and it has three steps. Step number 1 is carbon fixation. Step number 2 is reduction. Step number 3 is regeneration of RUBP. So let's focus one by one. So step number 1 is carbon fixation. And what is happening in carbon fixation? You see there is a 5-carbon compound called the ribulose bisphosphate. So ribulose bisphosphate, it's a 5-carbon compound, and this ribulose bisphosphate is actually reacting with carbon dioxide. So in other words, it is catching carbon dioxide, it is fixing carbon dioxide, and they're forming a 6-carbon compound. So let's see the figure, which will help us understand what's going on. So this is basically the Kelvin cycle, and we see in the Kelvin cycle, this is the ribulose bisphosphate, and why it is called. So ribulose means 5 carbon, 1, 2, 3, 4, 5. It's a 5-carbon compound, and it has 2 phosphate molecules. These phosphates are, now you see how this ribulose bisphosphate is capturing one carbon dioxide. This carbon dioxide is obviously coming from outside through the stroma, and how this whole reaction is catalyzed by the enzyme called Rubisco. Rubisco is the most abundant enzyme present in the whole biosphere. Okay, so Rubisco is the most abundant enzyme in the whole biosphere. What is biosphere? Biosphere means it comprises the atmosphere, hydrosphere, and lithosphere. All the organisms present in the lithosphere, hydrosphere, and atmosphere. Okay, so Rubisco is the most abundant enzyme present in the biosphere. So Rubisco is basically catalyzing this reaction, and it is basically acting like a scissor, because when these two react, they initially form a 6-carbon compound, because there is 5 carbon and 1 carbon, right? Carbon dioxide has 1 carbon. So 6-carbon compound is formed, which is split into PGA, or phosphoglyceric acid, or phosphoglycerate. It's an acid. Okay. So phosphoglycerate, or PGA. 3 PGA, because in the 3rd carbon, there is this phosphate group. All right. So this is called the phosphoglycerate. or phosphoglyceric acid. It is always in the ionic form inside the cell, right? That's why we call phosphoglycerate. So now, so this is called the first step is called the carbon fixation. You see the second step is called the reduction step, stage 2. And here we see the assimilatory power, ATP and NADPH, which were made during the light reactions. Look how they are required, how they are causing the reduction of 3-PGA into GA3P or glyceraldehyde, glyceraldehyde 3-phosphate, right? Again, the third carbon has the phosphate, so glyceraldehyde 3-phosphate. So if you in biochemistry, we know when an acid, something which has the COH, the carboxylic acid group, right? So this is a carboxylic acid, right? So when the carboxylic acid is getting reduced to an aldehyde, CHO group, this is called reduction. COOH, carboxylic acid, is reduced to a aldehyde. This is a reduction. So this GA3P, we'll see that 6 molecules of GA3P are formed. Out of that, 1 GA3P comes out of the Kelvin cycle. It comes out of the Kelvin cycle and it forms glucose. This ultimately forms glucose. This is the glucose that is made during photosynthesis reaction. The 5 GA3P, the remaining 5, they are actually regenerated. So this third step is called regeneration of RUBP. So they are regenerated to form ribulose bisphosphate. All right. So this is, you see how this is forming the glucose. 1 GA3P molecule comes out and it forms the glucose. Remaining 5 are going back to regenerate RUBP. So this third step is called regeneration of RUBP. So Kelvin cycle has 3 steps. We have the carbon fixation, where the RUBP is fixing the carbon dioxide with the help of the enzyme Rubisco. And then we have 3-PGAs formed, phosphoglycerate or phosphoglyceric acid. This 3-PGA is reduced by the assimilatory power ATP and NADPH to form the GA3P or glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate, 1 molecule comes out of the Kelvin cycle, for which ultimately forms the glucose. The remaining 5 are recycled to form the RUBP. Okay? So this is all about the Kelvin cycle. We will stop here. And in the next class, we'll do the part 2 of the photosynthesis, where we will talk about CAM, the crystallization acid metabolism. We'll talk about C4, photorespiration. We'll talk about the different experiments of photosynthesis. That will complete this whole chapter. So we'll stop here, and we'll meet me in my other classes where I'll discuss other topics. So bye-bye, and thank you for watching my lecture.